everyone. Today I will explain peanut bacterial wilt. Peanut bacterial wilt, commonly known as dead seeds and green disease, is an important peanut bacterial disease that occurs in a large area in many countries and regions. In the main peanut-producing regions of China, the incidence rate of peanut bacterial wilt is generally 10% till 30%, and can reach above 50% in severe fields, resulting in serious yield reduction and even no harvest. Peanut bacterial wilt is a typical vascular bundle disease, which can occur from the seedling stage to the harvest stage and is most likely to occur during the anthids. In the early stage of the disease, leaves are delayed to open in the morning and closed early in the evening. The first and second leaves on the top of the main stem in the above-ground part lose water and wilt first, and the top leaves of the lateral branches are dull and droopy, which can often recover at night and in the morning, but cannot recover in the morning and evening after 1 to 2 days. And the leaves of the whole plant or on one side wither and droop quickly. The chlorophyll of the withered leaves has not been destroyed. The leaves are dull and matte but still green, so the disease is called bacterial wilt. The underground part first turns brown from the tip of the main root, showing wet rot, and the root nodules are dark green, then expand upward, and finally the whole root rots. The fruit stalks and pods are also dark brown and wet rotten. The diseased peanut plant can be easily pulled up. When the roots or stems are dissected, the vascular bundles, especially the ducts, turn light brown to dark brown. Cut the tender diseased stems and squeeze them slightly with hands under moisturizing, and the stained white bacterial pus overflows on the section. This picture is a comparison of the stem base of the diseased plant and that of the healthy plant. Compared with the healthy plant on the right, it can be seen that the stem on the left has completely turned brown and wet rotten. The cells have completely died, and the plant has lost its vitality. Let's check the stem of peanuts after bacterial wilt. It can be seen that the stem tissue, especially the duct, turns brown and has lost its physiological function. If the diseased stem segment is immersed in clear water, smoke like turbid liquid can be seen gushing out from the cut, which is obviously different from fungal wilt, and is also a reliable basis for the diagnosis of the disease. The disease is a bacterial disease, and its pathogen is Solanaceae. The thallus is a short rod-shaped single cell with blunt round ends and 1 to 3 polar flagella without spores and capsules. It is gram-negative. The optimum temperature for pathogen growth and development is 10 to 40 degrees Celsius, and the optimum temperature is 28 to 33 degrees Celsius. The adaptive range of pathogens is pH 6 to 8, and the optimal pH is 6.6. Then, I will talk about the occurrence law of peanut bacterial wilt. The pathogen of peanut bacterial wilt mainly overwinters in the soil and can survive for 5 to 8 years. It can also survive in the residues of diseased plants, bacteria-carrying weeds, and manure mixed with diseased residues, and become the infection source at the beginning of the next year. Pathogenic bacteria are mainly transmitted by rainwater, irrigation, and agricultural activities. Pathogenic bacteria invade the host from the root wound and the natural orifice of the plant and enter the vascular bundle through the cortex. The pathogens multiply and spread rapidly in the vascular bundles, blocking the conduits and secreting virulent cords, causing plant poisoning, wilting, and green-orange symptoms. The pathogens can also enter the cell spaces between the cortex and the parenchyma of the medulla to cause disintegration and rot. Pathogens in rotted tissues can seep into the soil and spread through running water and other ways for secondary infection. The incubation period of the disease is 4 to 5 days. When the appropriate temperature is above 30 degrees Celsius, it generally takes 7 to 15 days from the onset of the disease to the death of the whole plant. In severe cases, the whole plant can wither and die in 2 to 3 days. The occurrence of the disease is mainly affected by climatic conditions, disease resistance of cultivars, and tillage cultivation. Bacterial wilt prefers high temperature and humidity. When the average air temperature is stable above 20 degrees Celsius in 10 days, the disease begins to occur. It is most likely to induce the disease when it is hot and rainy, especially when it is sunny and rainy, alternatively, or when it rains after drought for a long time and the soil temperature and humidity change sharply. Coarse sandy soil with shallow soil layer and poor water and fertilizer retention capacity, or yellow clay with poor permeability, and plots with many underground pests are conducive to the disease. The disease is more serious in continuous cropping fields than in rotating cropping fields, and it is easy to get diseased by excessive application of nitrogen fertilizer. Vine cultivars are more resistant to disease than straight cultivars and pearl bean cultivars. Southern cultivars are more resistant to disease than northern cultivars. Finally, let's summarize what we have learned today. Today, we mainly learned the symptoms, pathogens, and incidence law of peanut bacterial wilt. Please focus on mastering the symptoms and incidence law of peanut bacterial wilt. Okay, that's all for today. See you next time.